This recording is part 6B in the lectures covering the lymphatic system. 6A covered cell-mediated immunity, and well, this part is going to cover antibody or humoral immunity, which involves the B cells, but also it needs the help of the helper T cells, which were covered when I did the cell-mediated immunity, how we activated the helper T cell. Now, in order for B lymphocytes to kill off pathogens, they need to be first sensitized and then activated before they're able to ultimately indirectly kill off antigen-bearing substances by secreting antibodies. So over here on the far left, here is an inactive B cell. And what you have in the little red dots, those represent antigen. Okay, so the little red dots, an antigen. And B cells will go for things that are in tissue fluids, not infected cells. That's what T cells do. But you have this antigen, and what happens is it binds to these receptors. So you see kind of the kind of clear little Y's. Those are actually antibodies, which are receptors on that B cell. And the antigen will bind to it, so it's got to be specific. So we have these antigens bind to it, and at some point, it becomes sensitized, and you'll notice it transfers it to this right here. And what that is that's the blue is the class 2 MHC protein. So first we have a B cell that has become sensitized. Okay, there's an antigen that I recognize, but it's not going to do anything until a helper T cells tells it what to do. So it's just like a safety check. And so what we have over in the middle, we're going to have the activation. So here, this represents that sensitized B cell. Over here, this represents that helper T cell. And this helper T cell is activated. And that helper T cell, again, what we have is this right here is that CD4 marker, and over here there's that MHC class 2 protein with an antigen bound to it. And remember, anything with the CD4 marker can interact with class 2 MHC proteins. So we have a activated helper T cell, and right here is that receptor that's specific for that particular antigen. So we have a B cell that's sensitized. We have an activated helper T cells that says, yep, I'm activated because of that antigen. You need to start working. And so what it will do is it's going to stimulate division of those B cells and also cause the B cells to differentiate into these cells here, which are plasma cells. And so we're going to have a lot of cellular division. Again, you got some memory B cells, which you see here, but then the rest of them are going to divide, start to differentiate into plasma cells, and plasma cells secrete all these antibodies. And the B cell is going to indirectly kill off these pathogens by releasing antibodies. They don't like to get their hands dirty like the cytotoxic T cell. So they're going to release the antibodies, and these antibodies are going to end up causing the antigens to be, or should say, be destroyed. I don't know if that's the proper word I want, but we're going to look at how the different ways that these antibodies actually work. Now, I will post a YouTube video that kind of gives you an idea about the different types of, or about immunoglobulins in general, and then another one that goes over specific types of immunoglobulins. But immunoglobulins are antibodies. That's what immunoglobulins are. They have a specific structure. They have these heavy chains, they have these light chains. Pretty much the part that you see right here where it says antigen binding site, that's variable. And we have so many different permutations that you can have that we can have very specificities. This constant segment, what you have here, is that's what's going to give you the certain type. You have 5 types of immunoglobulins, and that's going to give its characteristics. Say, such an example, is it going to activate complement, where we're going to find it, that type of stuff. So it's going to give you the characteristics of that particular type of immunoglobulin. Now, if we kind of look at the classes of immunoglobulins, and again, you can watch a little video over it. There's 5 types: M, A, D, G, and E. And I know I went not in the order that you see here, but I remember this because it spells a woman's name, Madge. That's how I remember it. Is you have 5 classes of antibodies, and the ones that we talk about the most is the IgG and IgM. But obviously, these other ones are important. Now, IgE antibodies, they are important in response to I should say, perceived threat. So those are ones that are involved in those allergic reactions, like the type 1 hypersensitivity reaction that we'll talk about in a subsequent video. The ultimate purpose of IgE, not quite sure, but you find those in plasma and serum. They're a very small percentage of the total antibody population. They do not cross the placenta, and they do not activate complement. So they're important in just allergic reactions. There's a receptor that they have that binds to, they call it FC receptor, binds to mast cells and basophils, which trigger that release of histamine. Now, IgD antibodies are found on the surface of B cells. They do not cross placenta. They don't cross or don't activate complement. Now, IgA, these are ones that are found in the secretions from our body, like mucus and tears and saliva and semen. Colostrum is that first thing that's actually before breast milk is produced. It comes off as colostrum first. These do not cross the placenta. They don't activate complement, but IgA is important in protecting you. Really, one of the biggest things they protect you is against respiratory infections. And I actually had a student who had a specific deficiency in IgA type, and so she was more prone to having respiratory infections. IgG and IgM are the ones we talk about the most. You find those in plasma and tissue fluids, and both of those activate complement. That's kind of important. Now, IgG do cross the placenta, but IgM don't. So I got to mention this. So with that passive, naturally acquired passive immunity, which I had mentioned before, the kind that the baby gets from mom. This is not breastfeeding. Breast milk gives you that IgA, but with what mom confers some of her immunity to the baby because of the type IgG that cross the placenta. None of the other types cross the placenta. Now, I do want to mention is what is the difference between, so when we talk about blood types, the ABO and the Rh factor, is remember if a mother is Rh negative, she needs to be given RhoGAM shots to prevent any production of antibodies against the D antigen. Well, the Rh factor, or the D antigen, that crosses the placenta, and so that's an IgG type. But you don't have to worry about mom, if I'm type A, that I'm carrying a type B child, because the antigens for, or I should say, the antibodies against A and B do not cross the placenta, so those are actually IgM type. So it's kind of interesting. So IgG cross the placenta, but type IgM do not. Now we'll encounter, let me show you a picture first. Oops, sorry. The Ig, when you first have a, actually, I'm going to wait because I'm going to do the how antibodies work, and then I'm going to look at immune responses and look at IgG versus IgM. Now, how antibodies work is they're going to work a number of different ways. So one of the things that they can do is they can result in what we call precipitation or agglutination. So we actually have used that term agglutination before, as we talk about the agglutination that occurs when you're checking for blood types, is What happens is if you have a well with, say, an anti-A antibody in it, and the A antigen's present on that blood that you put in that well, and you mix it, you'll get clumping. And they refer to, in that case, agglutination. Well, what's happened is the antigen is on the cell membrane. So if I'm type A, I have A antigens on the cell membrane. If I have put B antibodies there, it will bind to it and cause a clumping type reaction. And so agglutination specifically refers to when you have cells starting to clump together, and it binds to antigens that are on the cell surface. Precipitation is a kind of similar type things, but in this case the antigens are soluble and they're not bound to antigens. Both of them are like when you have precipitation, things become insoluble. You have antigen antibody complexes that form, and they become so large they can't remain in solution and they precipitate out. But both of these, if you agglutinate or you precipitate, that will inactivate the antigens. Now, another way that we can inactivate a particular pathogen is by neutralization. So you mask a particular dangerous part of the antigen, so maybe it can't infect the cell, or you prevent it from, preventing from infecting a cell, or somehow you neutralize the biological effect of that particular pathogen. Another thing that antibodies do is they promote opsonization, and I should go here. Oop, nope. So I'm going to put opsonization. And what opsonization does, it makes complexes more susceptible for phagocytosis, so it coats things, kind of like complement did. So it opsonizes things, makes the phagocytes be able to engulf and eat these pathogens easier. It also activates complement. So we kind of have a double whammy. Complement does promote opsonization. Complement promotes phagocytosis. So I can go right over here, promotes phagocytosis. If you activate complement, it promotes the formation of the MHC complex, which leads to cell lysis. It promotes inflammation, as you see here. The antibodies also promote inflammation by promoting chemotaxis. So the chemotaxis attracts macrophage into the area and neutrophils, these phagocytic cells. So you have kind of an overlap with the specific immunity with the non-specific immunity. So antibodies are going to be doing things and promoting things that are part of the innate immunity. So antibodies are going to inactivate antigens by activating complement, attacking the antigens directly, which you kind of see right in here, stimulate local tissue changes that pretty much hinder these antigen bearing pathogens. Now, this last thing I want to talk about is your first encounter with an antigen versus the second encounter. So talk about primary and secondary immune responses. Is when you first encounter a pathogen, is the primary response is you initially see an increase of IgM type of antibodies. But shortly after, you start seeing a lot more IgG, and eventually they come down. Now, if you've been exposed to this pathogen a second time, what you'll notice is you see a large amount of IgG type that are going to be important in this one. Now, knowing this, it kind of can help us when we talk about if I go to the physician and they can actually measure IgM and IgG antibodies specifically for whatever they're going to, what you want to be tested for. They can determine if you're currently infected or you have a current infection, or they can tell if it's a past infection or if you're immune to something. So if they take, say, you're not sure, I've used this scenario in the past where a gentleman couldn't remember if he had the mumps. He didn't remember if he had a vaccine for the mumps, and his mother was passed, and his parents were dead, so he couldn't determine that. And an adult getting the mumps, especially adult male, is not good because it could actually lead to sterility. And so his child got the mumps, and so a lot of people is like, you need to go away, stay away from it so you don't get it. But he didn't know if he was immune to mumps or not. And then he started feeling ill, and he had the man flu. So he went to the doctor, and they did a test, and they could tell if he had the mumps, or if it wasn't the mumps, it was maybe he was immune to it. So they can measure the IgG and IgM type of antibodies for the mumps. Now, if the man had high levels of IgM, he's got the mumps. But if they tested him and he had high levels of IgG types, is that no, you've had the mumps before, or you've actually had the vaccine for it, you're immune from it. So in this scenario, he just had the man flu. He didn't have the mumps. His IgG levels were elevated. He'd been exposed to it because his child was sick with it, but he was not going to get sick with the mumps because he was actually immune to it. So they actually do this in real life. They can measure the IgG or IgM types of antibodies for specific antigens. So this is the last part of the antibody-mediated immunity, and include just the primary versus secondary immune responses.